All right, so thank you first to Will and Paul for organizing these seminars. It's great to have more touch points with this community outside of the annual meeting. And thank you very much for the invitation to present today and to all of you for coming. As mentioned, my name is Lisa Couper, and I'm currently a postdoc in environmental health sciences at UC Berkeley. And like many, or perhaps most of you, I'm really interested in how infectious disease systems are responding to various aspects of global change. And today I'll be speaking specifically about the potential and the evidence for mosquitoes to adapt to climate warming and what this may mean for mosquito-borne disease transmission in the future. And the theoretical underpinning here is that when populations are faced with rapid, stressful environmental changes, the heuristic in conservation biology is that there are broadly three types of responses: move, adapt, or die. So if we consider a population that's currently at the warm edge of the species distribution, under additional warming, it can move, meaning shift its distribution to track its current climate niche into the future. And typically, this would appear as distributional shifts towards the poles or upwards in altitude. Populations could also maintain their current warm edge distributions by adapting to warming. And here I'm using adaptation to encompass both evolution in physiological tolerance, like shifts in thermal performance curves, but also plastic and/or behavioral adaptive responses like shifting microhabitat usage or shifting the timing of seasonal activity patterns to minimize high temperature exposure. And then a failure to either move or adapt would lead to local extinctions. And I think understandably, there's been a relatively large focus on this first response, namely projecting shifts in vector or pathogen or disease distributions under climate change. And for mosquitoes and mosquito-borne diseases specifically, there have been a lot of modeling studies using various trait-based or statistical approaches to predict shifts in mosquito or mosquito-borne distributions under warming. And broadly, the consistent takeaway has been that in response to climate warming, mosquito-borne disease distributions are expected to shift polewards and upwards in altitude, specifically with expansions at current cool range edges and contractions at current warm range edges. But in a recent scoping review on mosquito vector range shifts that I was involved in, we found there has been documented evidence of expansions of mosquito populations at cool range edges that were explicitly linked to increasing temperatures. But we did not identify any studies to date, and this was just published a month ago, that have documented a contraction in mosquito populations specifically due to warming. And I think there are several possible explanations for this. One could be surveillance limitation, meaning it's harder to observe and confirm the absence of a species than the presence of it. And relatedly, it could be that warming-driven contractions are happening in places where there is not strong or consistent mosquito surveillance. So it could be happening and we're just not observing it. It could also be that contractions will lag warming by some amount, so they may be underway but not yet fully observable, essentially the concept of extinction debt. But another possible explanation is that mosquito populations are adapting in some way to tolerate higher temperatures than what we presumed to be their upper thermal limits. And it's this third possibility that I'm really fascinated in, and that's because mosquito adaptation to warming would mean that mosquito-borne disease distributions may be maintained in places where they're otherwise expected to decline due to warming. And therefore, we can't take our foot off the gas off of like vector control and disease control and prevention measures in those places. So there's some strong public health implications of this type of response. And so today I'd like to share three vignettes or studies related to the evidence and potential for mosquito climate adaptation. And they will focus on the following three questions. One, are mosquito populations currently locally adapted to temperature? 2, do mosquitoes have the evolutionary potential to adapt to warming? And 3, are mosquitoes using strategies to avoid heat stress? And I'd like to emphasize that these studies, which involved pretty large experiments and field surveys, were done with a large and fantastic team of folks, mostly at Stanford. And in particular, I want to highlight contributions from Mark Bitter, Kelsey Leiberger, Johanna Farner, and Aaron Mordecai, although many others contributed as well. Okay, starting with the first question, is there evidence that mosquitoes are currently locally adapted to temperature? And to investigate this question, we focused on this particular species, Aedes srensis, or the western treehole mosquito, and developed this as a model system for investigating mosquito climate adaptation. And there are a few relevant reasons why the treehole mosquito serves as an ideal model system. One is that it's abundant across a large climate and geographic gradient. Its species range spans roughly from Mexico to Alaska, meaning that if mosquito populations were locally adapted to temperature, we'd likely see evidence of it in this species. Secondly, across this distribution, it's present in large, spatially discrete populations. Namely, it completes it breeds and completes most of its life cycle in and around these individual water-filled tree holes, which are relatively easy to find and to sample from, and which form these nice natural meta populations. And lastly, it's congeneric with the major disease vector species, Aedes aegypti and Aedes albopictus. And it is a vector of dog heartworm, but it's not a vector of any known human disease, meaning that it's easy to manipulate in the lab without biosafety concern. So with this model system, we investigated the evidence for local thermal adaptation in mosquitoes using a common garden experiment. So here we first field collected populations from across most of the species range. We sampled from over 300 tree holes, which are denoted by the white circles on the map, and we focused on 10 populations for use in the experiment, which are the colored points on the map here. And these 10 populations covered a fairly large geographic and climate gradient. Namely, they spanned over 1,200 kilometers in distance and varied in their annual mean temperatures by over 7 degrees Celsius. We collected these populations as larvae and then reared them all in the lab for one generation under common temperature conditions. And this is to minimize any direct environmental effects on heat tolerance and to try to isolate genetically based differences. We then measured thermal performance across the mosquito life cycle on this F2 generation. And to do so, we randomly assigned the eggs from each of these 10 field collected populations into one of 6 constant temperature treatments, ranging from 5 to 32 Celsius. And then for each temperature treatment and population, we measured life history traits across the lifespan, namely larval survival and development rate, pupal survival and development rate, and adult lifespan. And in total, we had these temperature trait measurements on over 1,700 individuals from across 10 populations. And just to give you a sense for what the raw data from this kind of experiment looks like, it looks something like this, where we have measurements of life history trait performance here for larval development rate at discrete experimental temperatures. So here, each point represents a single mosquito from a single population. And for example, the yellow point here denotes the larval development rate of an individual mosquito at 28 Celsius. To that raw data, we then fit continuous thermal performance curves using a Bayesian approach, which allowed us to estimate parameters of the thermal performance curve, most of interest to us being the upper thermal limits, or the temperature at which trait performance drops to 0. And it allows us to estimate uncertainty in these parameters. And we repeated this process for each population and for each of the life history traits that I mentioned. And so what we're specifically looking for here when trying to understand whether mosquitoes are locally adapted to temperature is, one, is there variation between populations in their upper thermal limits, as seen in this schematic here? And if so, does it correspond to the source thermal environment, wherein populations from warmer environments have higher upper thermal limits than those from cooler environments? So is this the pattern that we found? And the short answer is yes, but only for one trait. Yeah, let me explain what's going on here. So I'm showing the fitted thermal performance curves for each of the life history traits that we measured. Again, larval and pupal development rates, larval and pupal survival, and adult lifespan. Here, each curve is the fitted thermal performance curve for a single population, and the curves are colored based on the latitude from which that population was collected. So warmer colors denote populations from lower latitudes, here warmer environments, and cooler colors denote populations from higher latitudes or cooler environments. And so the first thing you probably notice is that there's quite a lot of variation in heat tolerance between these different life history traits. However, what we're after is whether within a given trait there is variation in heat tolerance between populations. I think it's easier to see this if we zoom in on just the upper thermal limits for each trait. And so here I'm showing the point estimates and the 95% credible intervals for the upper thermal limits of each population. And as before, the populations are colored based on their latitude of collection, and now they're also ordered on the y-axis based on latitude. And what we see is that for the survival and the longevity related traits, there is no apparent variation in heat tolerance between populations. Namely, there's very little variation in the point estimates and or the credible intervals are overlapping for all populations. However, we do see variation between populations for the development rate traits. If we look first at larval development rate, we see there is clear variation between populations in their upper thermal limits. However, there's no clear pattern in this variation with the source thermal environment. That is, we see relatively high upper thermal limits for populations from warm, as well as intermediate, as well as cool environments. So there is intraspecific variation in heat tolerance, but it doesn't appear to be locally adaptive. However, if we look at pupal development rate, there are two findings I'd like to call out. One is that populations vary substantially in their heat tolerance for this trait. Namely, the upper thermal limits varied by about 4 degrees Celsius across populations, which for context is about 10 times the average amount of variation in upper thermal limits observed in other terrestrial ectotherm species when considering the same latitudinal extent. And that's based on this nice meta-analysis by Jennifer Sunday's group. So what this suggests is that mosquitoes have relatively high levels of standing variation in heat tolerance across their ranges, at least for these development rate traits. And the second finding I'd like to highlight is that variation in heat tolerance between populations does appear to correspond with the source thermal environment. And in particular, we see a general pattern wherein populations from warmer environments have higher upper thermal limits than those from cooler environments. But our collection sites did vary somewhat in elevation, so to make this finding more clear, we can instead regress the upper thermal limits of each population against temperature at that population's source environment. And in doing so, as you see here, we found a significant positive relationship, again, indicating that populations' heat tolerance is typically higher if it's coming from a hotter environment. And I've shown mean annual temperature as the metric of source environmental temperature here, but this held true when we considered other more specific, more biologically motivated temperature variables. So I think this provides strong evidence of local adaptation to temperature in mosquitoes, but why is it only observed in pupal development rates? Well, we think it may be because there are multiple strategies for coping with heat. For this species in particular, there's a diapause that occurs at the egg and larval life stages, and this could act to reduce individuals' exposure to high temperature stress. And then at the adult life stage, there's capacity for movement, which may allow individuals to seek out cooler microhabitats. However, at the pupal life stage, there are few of these alternative strategies for avoiding or coping with heat stress. Namely, there's no diapause, there's very minimal capacity for movement, given that these tree holes are not very big. And this life stage also occurs at a time of year when temperatures can be highly variable year to year and across space. So it may be that there's strong selection to advance rapidly through this life cycle, through this life stage in warm environments in order to minimize thermal stress. But to be clear, these are not conclusive explanations, but rather interesting hypotheses that this experiment generated and that motivated some of the follow-on work I'll describe later. So at this point, we really wanted to drill in further and understand what kind of evolutionary capacity a given mosquito population has to adapt to warming and what the underlying genetic architecture of mosquito heat tolerance looks like. And so I'd like to now transition to the second study, which was focused on understanding, do mosquitoes have the evolutionary potential to adapt to warming? And we first set about trying to understand how to even approach this question. And as it turns out, there's been decades of thinking about evolution and extinction in response to environmental change, mostly from a conservation biology lens. And the way this question is typically approached is to compare the projected rate of environmental change, for example, the projected rate of warming, to the maximum sustainable rate of evolution in the fitness relevant trait. So here, mosquito heat tolerance. And so you can think of this as the required amount of evolutionary change that's dictated by the environment and the maximum rate of evolutionary change that the population is capable of. If the projected rate of environmental change is greater than the maximum rate of evolution, we would expect populations to decline and eventually to go extinct. But if the maximum rate of evolution is greater than that of environmental change, then populations may be able to persist in place through evolutionary adaptation, which is a scenario called evolutionary rescue. And of course, the ultimate fate of a population will depend on additional biotic and abiotic factors, but this comparison at least provides a bounding box for what could occur. So how do we estimate the maximum sustainable rate of evolution? Well, we know from quantitative genetics that most simply this depends on several demographic and evolutionary properties of the population. Namely, it depends on the maximum population growth rate and the generation time, with higher growth rates and shorter generation times enabling faster rates of evolution. Depends on the strength of selection or how an organism's fitness varies based on its value for a particular trait, with stronger selection typically enabling faster adaptive evolution. And it depends on the genetic variance. So how much variation there is in the trait and what proportion of that is genetically based or heritable, with higher levels of genetic variance enabling faster rates of adaptive evolution. For mosquitoes, there were some estimates of various species population growth rates under different conditions, as well as their generation times, but there was little available information on the strength of selection on heat tolerance and on genetic variance in heat tolerance, meaning that at this time in 2021, we couldn't yet parameterize this kind of model and estimate the potential rate of evolution in mosquito heat tolerance. And so we designed a thermal selection experiment to address these knowledge gaps and to try to answer this question. And we again used the Aedes syrensis, or the Western treehole mosquito model system, for this study. And here we first obtained a diverse pool of starting individuals here from a single population of this species. And these individuals were collected as larvae and then reared under common lab conditions for 2 generations before use in the experiment. Then newly hatched F3 larvae were randomly designated into these replicated control or heat exposed groups, wherein the control group larvae were maintained at comfy room temperatures, while the heat exposed group were reared at 30 Celsius, which for this species is hot but not always lethal. Then all individuals that survived this artificial selection or the control conditions were reared out to the adult life stage, at which point we sequenced the whole genomes of each of these individuals. And we did repeat this whole experiment 3 times to obtain a total of a little over 100 individuals from each group. And I did want to mention that there was an additional piece of this experiment that I won't present on today for the sake of time, but prior to extracting DNA from these adults, we did conduct a separate assay of acute heat tolerance using a thermal knockdown assay. And we did also take a body size measurement approximation. So if you're interested in trade-offs in heat tolerance across the lifespan and relationships between body size and heat tolerance, feel free to ask about this, or we can chat offline about this another time. But for today, what I want to focus on is that this experimental design enabled us to estimate the strength of selection on mosquito heat tolerance. And this is based on the differential survival in the heat exposed versus the control group. And here we found that survival in the heat exposed group was a little less than half that of the control, indicating fairly strong selection. And this experiment allowed us to estimate or to identify the genetic basis of and variation in mosquito heat tolerance. And this is based on genomic differences in individuals that survived either the heat exposure or the control, or the selected or the unselected population. And I'll share the results regarding the genetic basis of mosquito heat tolerance first. So the first main takeaway is that we found that mosquito heat tolerance is highly polygenic, and I'll explain what I mean by that with this figure. So here I'm showing the genomic position of SNPs, or single nucleotide polymorphisms. These are specific points in the genome where the control and the heat exposed groups differ. So for example, the majority of the control group may have an A at this position of the genome, while the majority of the heat exposed group may have a T here. And the horizontal line is the threshold for statistical significance. So SNPs, or points above this line, are those that are significantly differentiated between the selected and the unselected groups. And so these are what we consider our candidate SNPs underlying mosquito heat tolerance. And we identified over 300 such SNPs across the 380 Syrensis chromosomes. So SNPs associated with heat tolerance are abundant and they're distributed across the genome, suggesting that many loci are involved in heat tolerance, or that heat tolerance is polygenic. And this is as expected. Traits like those involved in environmental tolerance and stress responses in ectotherms are often complex or polygenic. But despite being polygenic, there is also a clear clustering of these candidate SNPs. And in particular, using a clustering algorithm, we identified these distinct regions in the genome in which there was an elevated number of candidate SNPs associated with heat tolerance. And this suggested to us that there may actually be larger variation in the structure of the chromosome in these regions, such as insertions, duplications, or inversions, which are when segments of DNA are broken off and become reattached in the reverse orientation. And in brief, we used our short read sequence data to investigate the presence of these chromosomal inversions, which essentially appear as regions in your sequence data in which the orientation of the paired end reads differ from that of your reference genome. And in doing so, we found that inversions do appear to be present and abundant in the treehole mosquito genome, and they're roughly twice as abundant in these regions of elevated SNP signal, suggesting that they may play a role in heat tolerance in this species. And chromosomal inversions have previously been found to underlie ecological adaptation in many other ectotherm species, including in the malaria vector species. And the underlying mechanism here is that with inversions, you get lower recombination between the inversion breakpoints, effectively trapping a given block of DNA and enabling many potentially adaptive alleles to be inherited together. So even though heat tolerance is this complex polygenic trait, you can still get rapid evolution because these adaptive alleles are essentially being co-inherited due to the presence of that chromosomal inversion. And I'll just note that we're planning to follow up on this study using long read sequence data, which would allow us to more precisely identify the location of the inversion breakpoints and investigate their frequency in populations across the species range. But returning to the original goal of the study, which was to estimate the rate of evolution in mosquito heat tolerance, we now have the parameter estimates in place to do so. So that is, we now have an estimate for the strength of selection, again, the differential survival between the control and the heat exposed groups, and we have an estimate of the genetically based variation in heat tolerance from our sequence data. And again, from prior literature, we have estimates of maximum mosquito population growth rates under a range of settings, and we know the generation time for this mosquito species in nature is about a year. So we can now incorporate these estimates into that simple quantitative genetic model to estimate the maximum rate of evolution in mosquito heat tolerance. And we can compare this to the projected rate of warming across the species range to see whether or not populations have the capacity for evolutionary rescue or the ability to adapt on pace with warming. Specifically for our projected rate of warming, we were using the rate of warming in mean spring temperatures under a moderate climate scenario, as this to us seemed to be the most ecologically relevant metric of warming for this species. But we did consider alternative metrics as well as temperature scenarios or climate scenarios. And what we found in comparing these rates is that our estimated maximum rate of evolution in mosquito heat tolerance does exceed the projected rate of warming, suggesting that populations do have the potential to adapt on pace with climate warming. And specifically, the results I'm showing here are based on the point estimates or the average values for the parameters I mentioned, like the selection strength, genotypic variance, and population growth rates. But of course, we wanted to understand how varying these values might impact our interpretation here. And so we considered a range of values for these parameters using results from either a single round of the experiment as opposed to 3 rounds, or by varying population growth rates within a realistic range. And the maximum rates of evolution estimated under these alternative parameter values are shown in the gray lines here. And as you can see, under nearly all of these scenarios informed by our experimental and genomic data, the maximum rate of evolution does exceed the projected rate of warming. I do want to acknowledge, though, there are many simplifying assumptions underlying this model, including that there are no concurrent stressors or trade-offs to constrain rates of evolution in mosquito heat tolerance. Like there's no food limitation or drought or predation or parasitism, for example. We also assumed a constant strength of selection and genetic variance, although these could both decline either over time or over space, like could be lower for a range edge population, for example. And we haven't here incorporated the possibility for phenotypic plasticity or other strategies to cope with heat stress, which can modify the strength of selection and also modify population growth rates and therefore alter rates of evolution. And this is a non-exhaustive list of simplifying assumptions. So to be clear, what I'm hoping to convey here is that we found mosquitoes have the demographic and evolutionary potential to adapt on pace with warming. And whether or not a given mosquito population achieves this potential will depend on how some of these simplifying assumptions are borne out in nature. And I'd like to now hone in on this last assumption here regarding the role of phenotypic plasticity. And I'd like to do so by transitioning to the final study here, which was investigating the question, are mosquitoes using strategies to avoid heat stress? So not physiologically tolerating heat stress as I've been describing it, but avoiding it or minimizing exposure to high temperatures. And one key way through which mosquitoes, at least at the adult life stage, could avoid heat stress is through their behavior or movement. That is, adult mosquitoes are typically able to actively fly at least 50 meters, sometimes more depending on the species, enabling them to access various microhabitats for their egg laying and resting and host seeking activities. And these microhabitats can vary widely in their temperatures and their thermal regimes throughout the day. But of course, not all microhabitats are created equally, and mosquitoes have other constraints impacting where and when they can move. So what we don't know is whether mosquitoes in nature are indeed using behavior and actively seeking out microhabitats based on temperature as a means of minimizing their own heat stress. And this is called behavioral thermoregulation. And I think Erin Sawyer covered this really nicely in her EEID seminar last month. So the more specific question here is, does behavioral thermoregulation help mosquitoes avoid heat stress? And while the past two studies leveraged the tree hole mosquito model system and enables us to do these experiments we couldn't have done otherwise, here we took a computational approach to investigate this question at a more global level and for the mosquito vector species for which this question may matter the most. And so we focused on the 7 species listed here, which are the major vectors of dengue, malaria, or West Nile virus. And the overview of our approach is that we leverage this powerful suite of programs for biophysical modeling called Niche Mapper to estimate hourly mosquito body temperatures. Here, the first step is to use a mechanistically downscaled microclimate model to estimate microclimate conditions under varying levels of shade, ranging from full sun to full shade. These microclimate conditions are then used as the input to a biophysical model that estimates an organism's steady state body temperature. And this is based on those microclimate inputs, but also on user-defined functional traits like the organism's body size, its activity patterns, and its thermal limits. And critically for this investigation, whether or not the organism is able to behaviorally thermoregulate. So if this ability is enabled, then an organism would move to seek shade when nearing its upper thermal limit and move to seek sun when nearing its lower thermal limit, and its body temperature would be adjusted accordingly. We parameterize this biophysical model using prior estimates on these functional traits for each of the 7 major mosquito vector species that I listed previously. And we made these hourly body temperature estimates across a full year and across the known distributions for each species based on occurrence records from publicly available databases like GBIF. So in sum, what we have are these estimates of hourly mosquito body temperatures with and without behavioral thermoregulation across the year and across the distribution for seven different mosquito species. Lastly, to determine whether or not mosquitoes are experiencing heat stress at a given location, we use the framework of thermal safety margins from conservation biology. Here, you're comparing the organism's upper thermal limit to the hottest temperature it actually experiences. And so in this scenario where the organism's upper thermal limit is less than the hottest body temperature it experiences, that is considered heat stress or thermal danger. And the thermal safety margin in this example would be -3 degrees Celsius. Conversely, if the organism's upper thermal limit is greater than the hottest body temperature it experiences, that's considered thermal safety, and the thermal safety margin in this example would be +6 degrees Celsius. So what we're investigating is how behavioral thermoregulation impacts species thermal safety margins. And what we found is that behavioral thermoregulation does appear to be necessary for mosquitoes to avoid heat stress in their current ranges. And to explain what I mean by that, here I'm showing the fitted relationship between thermal safety margins and latitude across the range for a given species, here Aedes aegypti. Again, with positive thermal safety margins indicating thermal safety and negative thermal safety margins indicating heat stress. And I'm specifically showing here thermal safety margins estimated with behavior, meaning individuals have the ability to move and access fully shaded microhabitats. And as you can see, with behavioral thermoregulation, Aedes aegypti is typically thermally safe across its range. However, when thermal safety margins are estimated without behavioral thermoregulation, this species is in heat stress throughout the majority of its range. That is, if this species were restricted to fully exposed habitats, then it would likely be experiencing body temperatures above its own upper thermal limits throughout a large portion of its range. And because these estimated body temperatures are based on current temperature conditions in locations where the species has actually been observed recently, what that means is that behavior is likely already a common strategy for mosquitoes to avoid heat stress at the hottest times of the day and year. And I've shown results here for one species, Aedes aegypti, which is the main vector of dengue and is a relatively heat-tolerant mosquito. But these patterns were highly similar for the other vector species we investigated. And for visual clarity, I'm just showing the results for 2 other species, Anopheles gambii, which is the major malaria vector, and Culex pipiens, which is one of the West Nile vectors. Again, without the capacity for behavioral thermoregulation, species would be in heat stress, sometimes by several degrees across large portions of their ranges, suggesting again that in order to be consistently found in these locations, species are likely already using behavior to access shaded microhabitats during at least portions of their life cycles. And these model-based findings do match what has been observed in thermal gradient experiments in the lab, wherein mosquitoes behaviorally avoid temperatures above their critical thermal limits and spend more time at or just below their thermal optima. But while behavioral avoidance of high temperatures is likely a common phenomenon, what we don't yet know is whether there are fitness trade-offs in this strategy for mosquitoes and how behavioral responses potentially constrain or facilitate the evolution of physiological heat tolerance. So to summarize and synthesize the work I've shared today, we've found evidence that mosquitoes do have the capacity to adapt to climate warming, including through evolution in heat tolerance, as well as behavioral thermoregulation and potentially other evolutionary and/or plastic strategies. And why this is important from an applied public health perspective is that mosquito climate adaptation would enable these warm edge vector populations to be maintained where they're otherwise projected to decline due to high temperatures. Meaning we may be underestimating future mosquito-borne disease risk, particularly in the tropics, by considering only current heat tolerance in our projections of mosquito-borne disease. And so with that, I'd like to acknowledge the many co-authors and collaborators on this work, which included many vector control partners from districts across California. And thank you all for your attention and happy to take any questions you may have.
Thanks, Lisa.
Of course.
We'll open it up for questions. If people have a question, just feel free to unmute yourself and ask the question, or you can put it in the chat and I'll read it out, whichever you're most comfortable with. While we're waiting for people to muster up their questions, Lisa, one of my questions was not knowing much about the mosquito field. Has there been much on like epigenetic effects of heat in mosquitoes?
That's a good question. So how like heat exposure at one generation might impact future
Right.
generations' heat stress? Yeah, that's a really good question.
Presumably with your lab experiments, you're like potentially removing some of that by
Yeah.
mirroring them for the explicit purposes of identifying the heritable portion.
Exactly.
Right.
Yeah, like I was trying to control for those epigenetic or transgenerational effects as much as possible, although, we only reared them under common conditions for 1 or 2 generations. So some of these may have still slipped in. I'm not aware of any work looking into epigenetic effects. I, well, this isn't quite epigenetic, but I imagine there are maternal or transgenerational effects happening because high temperature exposure typically leads to faster development and then smaller body sizes, which can impact fecundity. So I assume there are these effects, at least in terms of like lower fecundity, but I don't know of how it might impact like the heat tolerance of the next offspring. That's a good question. I know there is Matt Thomas's lab at the University of Florida did this like multi-generational selection experiment in which I think it was Aedes aegypti they were working on, and they were passaging them for 10 generations, one at a control temperature and one at a kind of high temperature exposure. And they did find evidence of evolution and heat tolerance in that like 10 generation passaged line. So kind of similar to what I shared today, suggesting that mosquitoes do have that potential to evolve heat tolerance.
Other questions?
I actually had a question about the plasticity that you kind of raised in the second portion, and then also some of the results that you were showing in the very beginning with different TPCs for the different life stages. So if I remember in the first paper that you kind of talked about, there was really only a lot of like, potentially like strict, temperature gradient associated with that kind of like thermal max. But then there was no real variation with the kind of, a lot of the kind of like adult or like larval growth phases and stuff. Was there variation in, the size at which larvae were developing, too, in the different TPC conditions that you were showing, or like the size of the adults? You mentioned that kind of in the second chapter as like one of those mechanisms, but I'd love to hear if there's more.
Yeah,
more
that's
to
a
that kind of
really
story?
good question. Yeah, we unfortunately didn't take any body size measurements for that study, but certainly we do know that the higher latitude populations have just very obviously larger body sizes. Like it's just very clear when you're doing experiments on these populations that those like Oregon, Washington populations were much larger than the Southern California populations. I sorry, what was the second piece of your question? Like if in the experiment they were also developing into different body sizes based on their origin?
Yeah, just if you see different amounts of like plasticity in terms of response to temperature, there's different life stages.
Yeah, I mean, I guess so we did see for at least pupil development rates that the populations from warmer environments did develop faster at those higher temperatures. And that would mean that they did develop into smaller adults or smaller pupae. Um, yeah, some variation there, but yeah, we didn't quite capture like plastic responses in terms of like yeah their behavior or anything about like their like seasonal timing of activity. Although that would have been really interesting.
And then if you think about the third part where you're showing the basically like their behavioral ability to isolate from extreme temperatures,
Mm-hmm.
if you included that in the kind of like can their evolution outpace the speed of
Mm,
climate change, does that kind of like Bogart effect or whatever it is like
mm.
like actually make it so that it's less likely that the range edges are actually going to or the like the hot range edges are actually going to be able to keep up or keep pace with climate?
Yeah, it's a good question. Like I think yeah, I don't have a good answer to that from my work, but certainly plastic effects like behaviorally avoiding high temperature stress can then lower the strength of selection, so that can then reduce rates of evolution. It can be kind of compensated for by supporting higher population growth rates, so you're
Gotcha.
able to like
Yeah,
maintain a larger genetic pool, but the selection strength can be lower. So it's like it's not clear what the outcome of that is in terms of whether or not these plastic effects can constrain or facilitate evolution. But I mean, they act much more quickly, so it's probably my suspicion that those kind of behavioral shifts are more likely to be observable rather than like evolution, which we'll see kind of like more slowly over slightly longer time scales.
gotcha.
Yeah, good question.
I've got a question. Great talk, Lisa.
Thank you.
It's great to see you thinking through all the microenvironments and behavioral variation and biophysics, the sort of fine-grained aspects of temperature, and their effects on these functional outputs. I'm wondering, and sorry if I missed this, but how do you think about the timescales in terms of defining the thermal response scale? So, like,
Mm,
how long can they, like, you may show that they stop functioning at 33 Celsius or something, but, can they
mhm.
handle an hour at 35 or a day? And how do you integrate that kind of thinking? Looks like you're ready for this one.
Partially, yeah. So that's a great question, and I didn't explain this, but when we like in the thermal safety margins, we are specifically comparing upper thermal limits calculated from those constant temperature lab experiments, like what I was describing in the first study. So that captures pretty prolonged exposure to temperature stress, like several hours, several days. So then in the thermal safety margin estimate, though, we are just considering the hottest temperature that that mosquito experiences for a single hour, even. So we wanted to then investigate, okay, well, how long are they experiencing these stressfully high temperatures? And I'd say like here I'm just showing the consecutive hours in thermal danger or the consecutive days experiencing thermal danger, broken down by species or tropic, subtropic, temperate. And so roughly on average, like when they're experiencing thermal danger, it at most lasts for about 5 hours for most species, which at that time scale, it's quite possible that heat shock responses can kick in and be protective over those time scales, potentially still with some fitness loss. But those kind of heat stresses, on the scale of roughly like 2 to 6 hours, may be tolerable through again, these kind of like inducible heat shock responses, whereas, experiencing heat waves for many more hours than that and over consecutive days, I think that's where we'd really see like strong fitness effects. Mhm. Yeah, great
That's
question.
great to see you engaging with that one. Do you have any thoughts about, attacking that experimentally to see what the longer term implicate, or, how that plays out under
Yeah.
controlled circumstances?
I would really love to do that. So yeah, manipulating or yeah, imposing various lengths of like heat waves and seeing fitness responses to those. But I think another piece of that I'd really like to integrate is giving opportunity for repair. I think that's a piece that's also not often considered. So it's different if you're experiencing a 4 hour heat wave and then, straight to another like high temperature stressful situation. But if that's punctuated by cooler temperatures where mosquitoes are able to actively, repair themselves, I think the ultimate impact would be lower. So it just hasn't quite settled in my brain of like what the right experiment would be without needing to do like 30 different treatment conditions, varying the kind of spacing of the repair and the length of the heat waves. Although I think it's super important and increasingly so is like these extremes in temperature, the kind of hallmark of what we expect for climate warming.
Fantastic, thanks.
Yeah, thank you.
Other questions?
I have a question. So I thought that was a really interesting talk. Thank you so much. So in the first section, you kind of showed that, at least for some of the aspects that you looked at, it seems like heat and you can be evolved basically to handle heat in different ways. And so heat stress might affect these different things. And then in the second section, you talked about, you estimated standard genetic variation and strength of selection for, I think, just for the one mosquito species. So given kind of what you saw in the first section, like to what extent do you think that the estimates that you worked really hard to get for that second section are generalizable to other mosquito species? Do you think like not at all? Maybe maybe a lot, hopefully for you, I don't know.
Yeah, it's a great question. I would say like there have been several other studies now showing kind of similar pieces of evidence that mosquitoes have evolutionary capacity to adapt to warming. So I mentioned that Matt Thomas's group at University of Florida has found in these multi-generational experiments that Aedes aegypti can also evolve higher heat tolerance. So similar evidence in those major vector species. We've also seen Aedes albopictus rapidly shift its climate niche upon invading other continents. And we've also seen in Anopheles species, through the malaria vectors, reasonably rapid evolution given these chromosomal inversions. That's more in response to drought, but you might imagine that inversions again could facilitate rapid evolution of heat tolerance. So it's not a direct answer, but I imagine, other mosquito species have similar demographic and evolutionary properties to Aedes cyarensis, and that they can also have really large population growth rates. Most mosquito species actually probably have shorter generation times than Aedes cyarensis, which is a year, which is like rather slow. But what's kind of the missing piece is the extent of genetic variance in other mosquito species. That's what we really don't have a great sense of. We know there is some population variation in heat tolerance, and like some preliminary evidence of heritability and heat tolerance in other species, but that's sort of the missing piece from me giving you like a more complete answer of how these relate to other species.
Thank you.
Yeah, thanks.
Can you think of like malaria or dengue or these other, mosquito-borne diseases? Like I know there's work showing basically that like they can affect the TPC and they can affect these kinds of interactions between the mosquito and temperature. Does that vary across the kind of like life form or, I mean, like they're obviously acquiring the blood meal at different times and like the adult is, like I was just kind of wondering like if you're then thinking about how your results translate to like the potential vectoring of different diseases or how those diseases then affect like the mosquito phenotypes. Do you have any kind of ideas there or like experiments you're planning in the future?
Interesting. Yeah, so you're right. I have seen work finding that infection with some pathogen can impact the mosquito's thermal performance itself. And that, yeah, that would probably be most important at the adult life stage. And certainly, like, there is some vertical transmission of some mosquito-borne pathogens, meaning that they can be infected prior to their own blood meal. But I think it's generally, that's like generally less of like an important transmission pathway than horizontal transmission for mosquitoes, so like biting infected hosts at the adult life stage. So I do think like those impacts at the adult life stage are probably the most important. Yeah, I'll say too, like one of the main limiting factors mechanistically for transmission at high temperatures is adult mosquito survival, and particularly whether that's long enough for the mosquito, or sorry, for the pathogen to complete its um, uh, extrinsic incubation period. So like for malaria, for example, that EIP is often like 10 days. So if high temperature stress is constraining adult mosquito survival, meaning it's not able to live for those 10 days, which, you know, the malaria pathogen needs, then that's kind of like the limiting factor to transmission at high temperatures, whereas dengue's EIP is typically much quicker, like 4 to 6 days. So that's um, kind of a key aspect of like what's actually constraining transmission for both the mosquito and pathogen side at the hot end. I don't know if that answered your question.
Yeah, no, totally. Thank you.
Okay, maybe with that we'll call it good for the main session. If there are students that want to stay on the line, you're welcome to do that and ask Lisa additional questions. This is our experiment to see if other students want to talk in a less formal way afterwards. So feel free to like stay on. But for the rest of you, thanks for coming. And yeah, I'll just keep the line open for just a little bit longer if people want to ask anything that they didn't have an opportunity to ask about.